Welcome, everybody. Thank you, everybody, for coming today for joining us for the most recent iteration of the SIAH PGR Research Seminar Series. Today, we're very happy to have Beny Wagner, who's an SWW-DTP scholar who's working with us at Winchester School of Art. And he is going to be speaking to us today about his doctoral research, of course. And the title for his talk for today is Post-Industrial Metabolism and the Discorrelated Moving Image. Today, we're happy to have Jussi Parikka, who is Beny's primary supervisor, and he will be chairing the session. I'm just serving as the master of ceremonies and introducing Jussi and then Beny, and I'm going to disappear now and just listen and participate in the Q&A. Jussi, take it over.
Great. Thank you, Ryan, and hi, everyone. So nice to see, it's about to see so many familiar faces, but at least familiar names as it's, without video. And I'm super delighted to be sharing the session with Beny. My role is very minor, but I'll still sort of want to kick things off. And before letting Beny introduce and talk about the really fascinating topic about metabolism of moving images. As already mentioned, Ryan sort of gave an introduction already. Beny works, is doing the PhD at WSA at our university, obviously, and is also affiliated with the Archaeology of Media and Technology group. And besides all the scholarly work that we're going to be hearing about, one thing that I want to mention, and as you saw in the bio as well, is that I think Beny's work really nicely entangles with his artistic practice, including a very recent artistic practice that has been featured in multiple festivals in Europe and beyond as well. And very recently, Beny and Beny's collaborator Sasha Litvinseva finished and released a film, Constant, that is now, I believe, touring in different venues as well. And it's really one excellent recommended film to see if you have a chance. It's a sort of a really insightful take on history of measurement. I'll post a link on the chat so you can watch after the session, because that's slightly separate from, not entirely separate, but slightly separate from what we'll talk about today, but so that you know about the new cinematic work by Beny as well. But otherwise, just welcome, Beny, and we're really looking forward to hearing more about metabolism and images.
Thank you very much, Jussi. And yeah, and thanks to Ryan and Joe, who's not here, for organizing this this series and for inviting me to present. It's a really nice opportunity for me to kind of engage with everyone, because of course it's been like pretty limited with COVID, etc. So it's like a really nice opportunity. Yeah, I might just go straight. And yeah, I think Jussi posted the trailer for the film, and if anyone's interested, I can also send like a password protected link if you're interested in seeing the full film. And yeah, I'll just go ahead and share my screen and start. So is that's good?
Yeah, it looks good.
Great. Yeah, I took the kind of prompt to present something to share the kind of newest and actually like least processed aspect of the research so far. And I kind of, it was an opportunity for me to kind of try out how it sounds. And so I've kind of put this together today. Yeah, I look forward to hearing kind of what works and what doesn't, and kind of get a sense of how to keep forming this into the chapter that I'm working on at the moment. And so the working title for the PhD is Metabolism of Moving Image, as Jussi mentioned. And the basic idea is fairly simple. I'm making an argument for why there are several reasons to study the respective evolutions of moving image media and metabolism together. And to summarize very briefly, both of them are grounded in the 19th century in industrialization. Both have histories that run adjacent to each other. Both kind of radically reconceived the ontologies of bodies and how we think of motion, energy, matter, and both have been central to the reconfiguration of the modern self. First, kind of in the modern understanding of self as kind of an individual, a bounded individual, and then more recently in the last decades. I mean, I guess it's hard to say exactly time frame, but I would say both moving image and metabolism have been really central to reconceiving of the human as decentralized and distributed, in much more so than kind of previously imagined. And I guess it's actually that aspect that I'm going to be talking about most today. And just the kind of basic background to the project as a whole, I came to these ideas kind of initially grew out of my practice as an artist and filmmaker, and certain questions that I had that came up in relation to filmmaking as a tool that kind of like, on the one hand, it creates new boundaries between things, and on the other hand kind of transgresses them at the most basic level. And I wanted to kind of think about and act, can create a historical and theoretical practical framework to work with moving image as something that is deeply interwoven in the world, and to think of filmmaking as a kind of immersive way of being in the world, and as Thomas Elsaesser really nicely says. And these are a lot kind of some of the titles that were influential to developing my view of filmmaking, and especially the relationship of filmmaking to the body, to notions of life, and to thinking about film or moving image more generally as a kind of threshold between the body and the world. And so it was initially pretty open and maybe possibly kind of metaphorical sense. I came to this idea of moving image as a kind of metabolism. And in this kind of early and uneducated idea of metabolism, I thought of it as a kind of moving threshold between the self and the world. And but even at that kind of early stage, my lack of knowledge of what metabolism actually was, this was my big question at the beginning, but like, okay, I have a kind of intuitive sense of what metabolism is, but what is it actually? Like, I don't actually know what metabolism is. And I had some problems around that, like how is metabolism the same thing, like that's happening in my digestive tract, and the same thing that's happening in a single cell cellular organism, and at the same time what's happening on the kind of scale of the ecosystem. It seemed that metabolism was used in all these ways that couldn't possibly be really the same thing. And so as a non-scientist, for me to grasp kind of a complex scientific concept like this, the only real way for me was to kind of try to understand it historically. So to look at the social and political technological processes that led to its emergence. And so the project in that sense kind of is the archaeology of metabolism. And here, I mean, I guess this is like this, you know, on the one hand, on the left, we have this very kind of classic definition of metabolism that seemed to me initially kind of way too broad. I didn't feel like I got closer to kind of really what metabolism was. And then on the other side is like a chart of metabolic pathways, which is so complex and impenetrable to me as a non-scientist. And neither of these kind of situate what metabolism is. They don't place it in history. I know, for example, that on the thing on the right could not have existed in I mean, not even in the 18th or even really 19th century, but I don't understand what initially didn't understand what differentiated that from earlier models of the body. And
can I just briefly ask, can you make the slides larger as well so that it's full screen? At the moment it's showing it's tiny bit small. It's like probably in presenter view, just there's a comment. Also, there was a comment as well that if there's a possibility of turning on caption for the meeting. I don't know.
Oh yeah. How does that?
I don't know if that should be done by the host. Sorry to interrupt, but it might be
live,
in case
I have there's like live transcript, request host to enable live.
Yeah.
Yeah, I'm not sure if Claire can do that or not.
Just so they know, but if you can make the slides large.
With my mouse, I'm gonna like this. Hold on. I'm gonna exit this for a second and do it like this.
Yeah, I think that could work because this way it might do it.
Oh, that's like a view. I don't know if that's really disable. I've never done this before. Show subtitle. How is that? Can you see me? Yeah, that's working, right?
Yeah, I think the captions are on, and
yeah,
just so that the image slides are big as well. Okay, thank you.
Okay, yeah. Let me see. Yeah. How is this now? In terms of image size?
It's just gone back to the same slide presentation mode. Yeah. I think that you might just, this would be even bigger.
Is this better?
Yeah,
this is even bigger as well. So perhaps keep this if this.
Okay, okay, maybe we'll keep this.
Yeah, okay.
Yeah, so I guess about, so this was kind of my starting point, and I guess around 2/3 into my first year, I came across the work of the historian of science, Hannah Landecker. I have some titles here at the bottom that you can see. And this was kind of really like a major resource, a really important milestone somehow in my process. Because she really has the incredible skill, I would say, of starting from the hard experimental science and working outwards. And she's really committed to thinking about the significance of metabolism broadly in the history of European science and industry, and can translate those things in a way that someone like me can understand. Probably someone like anyone here is listening. So the main point is which I'll get to, is that there's a profoundly historical relation of metabolism. Metabolism is not something that is just obvious or given. It's an intensely historical process. And so today I'm going to talk about very contemporary processes. The title is like post-industrial metabolism, discorrelated moving images. And but I wanted to give this very brief background to the project as a whole, because it's actually mostly historical, and I'm kind of tracing an arc from the late 19th century to the present, and to kind of build these points of convergence between metabolism and moving images, and to demonstrate how they're both dynamic systems that are constantly changing. And I refer to metabolism as a system. That's, there's many ways to think of metabolism, and any way, I guess, that you would think about it is inevitably partial and limited. And so to call it a system it's with the recognition that it's not only always a system, but in the way that I'm thinking about it in this, especially here, it is as a system, and to think of both moving image and metabolism as systems that are designed to measure change, but that are constantly changing themselves. So on the one hand, they're both removed from the world because they create models of things. They create models of change and the models of the world in motion. But as models themselves, they're constantly changing. And so in that sense, they're also a part of the world, and they're always kind of operating in this duality, this dual tension. Now the only thing is, here, okay. So in 2017, a group of scientists led by Seth Shipman, working at the intersection of molecular biology and neuroscience at the Harvard Medical School, published an article in Nature detailing the results of their experiment, where they used the CRISPR-Cas system to encode a moving image into the DNA of a living bacteria. The images they chose for the experiment were none other than Eadweard Muybridge's iconic sequence of photographic stills from 1879, capturing the horse NEG in full gallop. And these, I think these particular images were taken to resolve a kind of argument that people had about whether or not a horse lifted off completely from the ground when it was galloping, or whether one of its legs stayed on the ground. But aside from solving this kind of relatively insignificant seeming argument, they were also the kind of often considered to be kind of one of the foundations for cinematographic motion. And this kind of decision to kind of reappropriate these images and literally encode them into the cell of a living bacteria was, of course, very reflexive. Here, this is a quote from one of the people involved in the experiment, George Church, who runs what's called the Church Lab at Harvard Medical School. And he says a horse galloping is biological information over time, and it's one of the first examples of recording any kind of motion, especially biological motion. I don't know that this is necessarily true in terms of the first. I think it's maybe a limiting perspective, and I think also both UC and Ryan also have know a lot about the various ways of recording motion, and there's a lot of how we define motion and biological motion and recording has as much to do with how we define where something begins and ends. So not exactly sure, but nevertheless, the point is that there's a kind of, they're placing themselves in a lineage very intentionally. And yeah, and so the article in Nature is a step-by-step breakdown of how of the experiment, and I'll talk about that in some more detail. But mostly it's written in technical terms that are difficult to grasp fully without specialist knowledge. But the story still became very popular, and it was reported in many major media outlets. And many of the reports featured this reconstructed GIF of the 5 frames. It's 5 still frames set in motion. And on the left, you see the original images that were delivered to the bacteria. And on the right, you see the reconstructed images that were derived from sequencing the replicated cell's genome. And so they're, of course, incredibly kind of close to the original, but they have these stray pixels, which you can see on the right. And I'm pretty interested in these stray pixels because so the researchers describe the 90% efficiency of their experiment. So that refers to their ability to accurately retrieve the data stored in the genetic sequences. But at the same time, for a human observer, even one with specialist knowledge, these stray pixels are the only sign that anything happened. Like otherwise you wouldn't, like without the story, without the backstory, these images are actually kind of impenetrable to us. And these stray pixels that could have been made with in all kinds of different ways, but they tell us something happened. They tell us something has changed. Yeah, so I think so. Just to kind of and I should also preface this by, I'm a little bit out of my depth, or quite a lot out of my depth dealing with molecular science. So it's not something that I'm trained in. I'm very interested in these images from a social, cultural, technological perspective. And so, I feel like it's important to, I've been trying to understand how they're made and understand the dynamics of how they're made, but it's not something that I'm in any way really qualified to be talking about, but I'm explaining them in the context of the interest of these images as objects in the world. Seth Shipman, in an interview, wrote, "DNA is an excellent medium for archiving data," and these images, Muybridge images, are not by any means the first data to be stored and retrieved from DNA. So it's for about 2 decades. There have been multiple projects that have delivered data into living cells. And actually the idea that data could be used as a storage, that DNA could be used as a storage device dates back to the discovery of the double helix in the 1950s, and Richard Feynman, who is like a really interesting physicist. I recommend watching videos of him talking. They're quite a character. He would imagine the kind of vast amount of space that would be opened up if we had the technological ability to store data in DNA. But it wasn't really until computation was powerful enough that it became possible to sequence and edit genes in a concrete and targeted way. And CRISPR is a kind of genetic memory. It's a bacteria's own genetic immune defense system. And for now, for just over a decade, it's been possible to edit this memory using the CRISPR-Cas system. And the system is fueled by increasing accessibility of resource-intensive computation, and that was previously very costly. And it's leading to what many are calling a new era of genetic engineering, as well as a new gold rush in biotech investment. But on a very basic level, the CRISPR-Cas system allows people to locate a specific sequence of DNA within a cell and replicate a given sequence with something else. So this means that essentially any data that can be converted into binary code can be it can be converted into the AGCT nucleobases that makes up the DNA's double helix. And this is a graph that shows, that was published in the Nature article, that shows exactly how they broke the image down into its pixel values and encoded that into the nucleotides of the DNA. And what they did is they distributed, and this is kind of important in terms of like my material understanding of this and its broader implications for digital media, is that they distributed the data across approximately 100 different genetic strands. And these strands were fed into living bacteria, and these living bacteria incorporate the strand into their own genome. And that means that every time a cell replicates, it's replicating this data. And yeah, and so what happens, I mean, I guess what we could, well, I'll talk about that later. Yeah. This idea that DNA can be used to store media was taken up quite excitedly, and many of the articles I encountered, and people kind of many authors imagined kind of this like new horizon that would be kind of opened up, where we could have data centers. And here's a kind of graph that shows how, if a single gram of DNA can store 100 billion or 1 billion terabytes, there's a kind of essentially limitless horizon, because it's seen in a very abstract, scalable way. And people imagine kind of like how we could provide solutions to the various burdens concerning space and material resources, energy inputs, waste outputs that are such a threat to data maintenance at the moment, and carrier obsolescence. And there's a kind of, I would say, media archaeological vernacular in these kind of public discussions. But the lead scientist in the project is very clear that he's not really interested, or the point of his work isn't so much to use DNA as a storage device, but rather to create a system where a cell could record data from its surrounding over time. And he says, you know, we want to turn cells into historians. And he's a neuroscientist, and he kind of explains how limited knowledge is of the brain, because the fundamental problem of observing neurological activity is that the techniques of observation disrupt the actual activity. And so any observation is inevitably quite partial. And if a cell could be inserted into the brain and could be what's kind of what he refers to as molecular recorder, it could collect data about neurological activity without interfering in the processes. So this, of course, is just an idea, because the experiment involved externally delivering DNA strands in a controlled environment. So it's not actually, you know, the cell isn't operating on its own volition, I guess. But I was interested in this idea because it kind of taps into like a fundamental problem of both scientific observation and cinematic recording, and particularly the way where these two things intersect historically, and it kind of centers on this question of how can we record something without altering it in the process, and how can we know something without our knowledge becoming glued to it or destroying it in the process, or altering it in the process. And you know, this is I come from a perspective where I consider this as a philosophical problem, and it's a philosophical kind of problem that's associated with people like Georges Canguilhem and Gaston Bachelard and Bruno Latour, Evelyn Fox Keller, Donna Haraway, Karen Barad. But in Shipman's approach and the approach taken by this lab, it seems like it's a strictly technical problem. And it's not so much that the observation inevitably creates an abstract model that separates from the thing observed, but that we simply haven't created tools of observation that are sufficiently embedded in the processes they observe. So, you know, in a sense, there's kind of this divide of between the kind of an understanding, let's say, that unknowability is inevitable, and that versus a view that thinks kind of that everything is knowable. It's just that we don't have the right tools. Like everything can be knowable with the right tools. And Georges Canguilhem writes about this in depth and over a long period of time. And it could be kind of considered in a vitalist mechanist lens, which is a couple of centuries old, I would say. So of course I can't resolve this debate in any way, but I'm interested. So I guess when I started this project, I never thought that I would encounter something that existed that was literally like a metabolic moving image, or in a way that was so literal as this. Like there's literally an image in a cell, and it's working within the cell's temporality, and of course, as I said at the beginning, I'm still kind of working through this material. You know, for me, it's a kind of this experiment acts as a concrete prism to think about the broader issues that I'm investigating at the intersection of the models of life and our understanding of life, image making technologies, and how these are kind of historically really interwoven. And so this issue of integration, to have a cell that has recording capacities that can inhabit a space without interrupting its operations, is kind of this dream of recording that's fully integrated into its environment. And that's, of course, what I'm interested in thinking about, this immersion. And I think this experiment shows that that kind of immersion is an issue of scale. On the scale of our bodies, like the scale that we spend, all of our sensual, our sensation, sensorial experience on, things seem distinct. So my body is distinct from a camera, is distinct from a screen, is distinct from a server, and so we need we need complex ideas that show us how those things are interwoven. And of course, UC has worked on this, and in a lot of on the scale of on the planetary scale, that immersion is very clear. On a long-term temporal scale, it's very clear that we're immersed in our tools of observation, and that they're not distinct from us. And this kind of idea of a molecular recorder on the molecular scale also kind of shows this idea of perfect immersion. Yeah. So I'm trying to kind of think like in terms of time, because now it's I'm just trying to decide what level of depth to go into.
Yeah, we should leave time for questions. So basically, if we think that it's now 35 past, that, if another 10 minutes or so, would that be okay? And then it would leave at least 15 for discussion.
Yeah, I'll do my best.
Yeah.
Yeah, I think I'm maybe about halfway through, so that's probably unrealistic. And so I'm gonna kind of, race through this. I think I'm probably not going to be able to get to the issue of discorrelated images. I think I'm mostly just going to talk about metabolism. But yeah, so I'm but I guess the idea of this chapter that I'm working on is that I use this experiment as a kind of object to that I unpack it through kind of two theories. And one is Shane Benson's notion of discorrelation, which is a concept he's developed to try to map the kind of changes brought about by digitally networked media in what's often referred to as the post-cinema. So it's a theory that emerges from a media theoretical space, and but is very interested in embodiment, and is very interested in the exchange between the body and digital media. So that's one way to look at this experiment, and the other is through Hannah Landecker's mapping of the changes in the metabolic sciences over the last century and a half, but particularly what she's identified as a condition of metabolic sciences in the last several decades, which she refers to as post-industrial metabolism. And to understand what post-industrial metabolism is, it is necessary to kind of backtrack. This, I have a whole chapter essentially on in the industrial model of metabolism in relation to the emergence of early cinema. And I was going to go into that a bit here because it's kind of maybe important for context, but I don't think I'll have enough time to. Maybe suffice it to say that the model of modern metabolism that is still very much what I think we think about when we think about metabolism is still very much a product of this industrial model derived from the middle and late 19th century, which modeled both on the level of on the scale of the body, of the human body, and on the scale of the cell. It was very much modeled through on the industrial factory and on industrial tools like the combustion engine, and yeah. So in contrast to that, so I'm sorry that was like very brief, but I'm just trying to get to post-industrial metabolism, which is which Hannah Lendecker understands as a regulatory zone. These are two quotes from her. It's no longer this factory system modeled on very distinct ideas of matter and energy, and on like a motor, but it's much more. We could understand it much more as a network of mediation between the body and technical society. So it's no longer, what is this? How does man and how do man and nature intersect? There's a kind of understanding that the environment has been turned into a kind of technical interface, and we're regulating almost kind of like in the way that we could think of maybe a data center with cellular signals, and that's really how metabolism is explored in the current models. Pass through that. Yeah, and so this is kind of a quote that I really like from Lendecker's work. That is where she writes, "Metabolism is not a concept floating above time, influenced by metaphor, imprinted by context, more than a shift from one theory to another that could be described as a history of metabolism. Metabolism is in history." And so when she refers to a post-industrial metabolism, metabolism is both studied through the post-industrial systems of knowledge and systems of mediation, but it's also that the industrial model of metabolism has literally changed metabolism. So it's literally changed kind of not only what we know about organisms, but what organisms are. And I guess one of the things that I'm interested in this is how these models create different models of subjectivity, how we understand ourselves and our relation to the world. And we can think about this post-industrial model of metabolism also unfolding against the backdrop of very new models of life that have emerged in recent decades. And there's many ways to think about that. One would be like Lynn Margulis's model of the holobiont, which accounts for a host organism in relation to all the microorganisms that it's immersed in. That concept has been expanded in the concept of the hologenome. So to think about on the genetic level, how anything we could think of as a biological individual is composed of many overlapping and distributed parts. And so this kind of the model of the body that emerged from industrial era metabolism as this kind of enclosed. I didn't talk about it before, but it was very much modeled on the enclosed cell and the membrane as something that distinguishes the inside from the outside, creates a kind of hard boundary around biological individuals, has changed very dramatically through these other models of life, and has changed very dramatically how we think of ourselves. This is very much present. This kind of post-industrial model of metabolism is, of course, very present in the Muybridge experiment, the Muybridge DNA experiment, because there's this kind of fascinating inversion of interior and exterior and temporality and scale in a way that destabilizes kind of classic divisions of subject and object. We have these images taken in the late 19th century that measured the movement of bodies externally, and these are literally incorporated into the cell's interior. And the cell's own temporality is repurposed to produce cinematographically reconstructed temporality of the horse. And I have here two quotes by Deborah Levitt, who wrote this great book called The Animatic Apparatus in 2018. And she has some really nice observations along what she calls the mediology of life and the life of media in terms of articulating kind of how closely interwoven our models of life are with our image making technologies. And one of the things, where is this? Yeah, so she has, so she writes, we're cinema and the cinematic biopolitical regime. So here she's referring to classic cinema, the 20th century model of the cinema is structured by a distance between the screen body and the spectator's body. The animatic apparatus, which is the current media, digitally networked media images, she writes, always tends towards merging and exchanging image space and body space. So kind of reconfiguring what the separation is between the body and the screen, between the apparatus of capture and bodies in space. How are we? I mean, we're now at quarter of.
Yeah, I mean, if you can start to wrap up with a conclusion that we can then sort of follow up with questions and continue the discussion in the QA mode.
Yeah. So I can't kind of sufficiently go into kind of Shane Benson's idea of discorrelation on a very basic. So there's many ways that he uses discorrelation to think about the kind of new subjectivity that emerges between the digitally encoded images and the spectator. But what I guess what I do in unpacking this idea of discorrelation in this work that I'm doing now is that I want to kind of propose that discorrelation doesn't only refer to the media themselves and the spectatorship that emerges, the type of spectator that emerges through that media, but that this discorrelation is a kind of like a new ontology of life and of living systems. And in this is kind of where I'm beginning to formulate these ideas around the Muybridge experiment, is that what we're seeing in this experiment is a reconfiguration both of kind of the living system and how it's mediated, and in that kind of mutual reconfiguration, we see how closely intertwined they are. And yeah, I'm sorry, I feel like I haven't really given enough background to really ground these ideas, because I think I overestimated what I could get through, but hopefully we can discuss that a bit if people are interested.
Brilliant, Beny. Thank you so much. I think you gave already a lot for us to think about and ask questions about as well. So as mentioned, we have time. So please, and I wrote in chat as well that people can post questions both in the chat by writing there if you feel that more comfortable with that, or then you can just jump into our discussion in voice as well. We've been in dialogue with Beny for a long time about this, so I just really want to prioritize everybody else in terms of Q&A. We have the first question as well, and there's no such thing as a stupid questions.